Nice to meet you everyone. This is Freddie Yun, who is a technical sales manager in CJ Bio. Today, I would like to introduce the CJ Bio's new product Nextide. Nextide means next generation of novel protein source for the monogastric animals that efficiently replace the high quality animal protein source. The contents are as follows: a brief introduction, and then the protein source that we are using in the base ingredients. Thirdly, the functional amino acid and nucleotide blend that we used in this product. Finally, the animal trial with economic profit of the Nextide. This page shows the summary of our new product. As you can see in the left side, that we are using low molecular weight peptide as a base protein source. It is Bacillus fermented soybean meal that developed by CJ Bio's own R&D technology. As a single source of Bacillus fermented soybean meal, it has low anti-nutritional factors and highly digestible, as well as include probiotics. However, compared to fermented soybean meal with animal protein, such as plasma protein and fish meal, it has relatively lower functionalities. So, we added functional amino acid that enhancing the mucosal immunity and antioxidant capacity and block inflammatory cell signaling. Finally, we added nucleotide because when we replace the animal protein by plant protein, there is a big difference of nucleotide contents that can accelerate epithelial cell regeneration, some of prebiotic effect, and increase the feed palatability. Including all these functionalities, we believe that the Nextide is new economic functional protein that replace the animal protein efficiently. When we compare the safety of the ingredients, the major animal source, fish meal, has a lot of heavy metals and higher ash contents compared to Nextide. Therefore, to ensure consistent quality to meet regulatory compliance and for economic values of the product, we should be considered alternative source to reduce the risk. Soybean meal is a good plant protein source. However, it has limitation, especially for young animals, because of higher anti-nutritional factors like a trypsin inhibitor, allergens, and non-digestible carbohydrates. So, various methods has been tried to overcome these issues, and CJ has adopted the Bacillus fermented process. Let's talk more details. From the nature, soybean fermented foods such as Korean doenjang and Japanese natto are naturally fermented by Bacillus strain. As you can see in left side, Bacillus can produce higher enzyme activity of carbohydrates and protease, and it has faster growth rate. So during the fermentation, that higher enzyme activities can efficiently remove the anti-nutritional factors. However, most of the competitors are using lactobacillus as a soybean meal fermentation. There is a lower efficiency of the enzyme activities and growth rate. As you can see in the right side, because lactobacillus are main strain for dairy product like a yogurt and cabbage product like a Korean kimchi. So, lactobacillus is not suitable for soybean meal fermentation. As you can see in this electrophoresis data, soybean meal, lacto-fermented soybean meal, and enzyme-treated soybean meal have still allergen and trypsin inhibitor bands. However, Bacillus fermentation soybean meal from CJ Bio showed that the allergen and trypsin inhibitor bands are degraded into the low molecular peptides. Due to the strong protease activity during the fermentation process, almost completely minimized anti-nutritional factors. Seen here, the most of the lacto-fermented soybean meal products in the market have only 30 to 40% of low molecular peptides. However, Bacillus fermented soybean meal showed more than 60% of low molecular weight peptides. Therefore, better performance can be expected in animals by using Bacillus fermented soybean meal. In case of raffinose and stachyose, which are non-digestible carbohydrate fractions, especially galacto-oligosaccharides, as you can see, the data which analyzed by HPLC machine. The peaks of the raffinose and stachyose are very high in soybean meal and lacto-fermented soybean meal. However, bacillus fermented soybean meal almost eliminated. Additional benefit of the bacillus fermented soybean meal is probiotic effect. The fermented strain bacillus is super thermostable. You can see in left side, when increase the temperature up to 100 degrees Celsius, there is no reduction of the bacillus counts. And see the right side that did extruding test with 10% inclusion of fermented soybean meal in the shrimp diet. Up to 110 degrees Celsius, there are no reduction of bacillus counts. Normally, the pelleting temperature is not over than 95 degrees Celsius in swine and poultry diet. So, bacillus can survive and it goes to intestines of the animals and working as a probiotics. As a probiotic effect, we tested antibacterial activity against major pathogenic bacteria such as E. coli and Salmonella in livestock and Vibrio and Aeromonas in aquaculture. We added about 10 to the 5 to 6 bacillus culture medium to the flask that is growing these pathogens. As you can see, on the left side, the pathogenic bacteria cannot grow around the bacillus culture medium, which means it has a strong antibacterial activity. And on the right side, we also tested the quorum quenching effect. If you look at the negative control, where distilled water was added, it showed a blue color, indicating that quorum chemical produced by pathogenic bacteria. This means the pathogenic bacteria were grown. And you can see CJ Bacillus treat 1, 2, 3, 4 showed that the quorum chemical was significantly reduced, similar with the positive control that antibiotics were added. This indicates that CJ Bacillus has strong antibacterial activities. From this slide, I would like to introduce the functional blend that optimizes amino acids and nucleotides. Nextide is product blended with a functional amino acid and nucleotides in appropriate amounts. It performs various essential physiological functions as shown in the table on the right. In cases when animals are infected with various pathogens, antibiotic-free diet, the not good condition of farming environment and the health of the livestock, or when dealing with the young animals, the demands of these functional amino acids increase because they play a crucial role in immune activities. All these amino acids are essential for growth, and their requirement has been determined under normal animal conditions. However, if these amino acids are utilized in immune responses due to the various reasons, growth may be depressed. So, to address these issues, the contents of essential amino acids has been supplemented. This diagram illustrates the cell signaling molecules related to inflammation in animals. The arrows indicate promotion, and the black bar represents inhibition. Functional amino acid promoting the expression of anti-inflammatory signaling molecules, while inhibit the expression of inflammatory signaling molecules. For example, arginine promotes the synthesis of the nitric oxide, which suppress the inflammation. And threonine, methionine, and arginine inhibit the production of nuclear factor kappa B, a factor that induces inflammation, thereby minimizing the inflammatory responses. So, proper blending of these amino acids and supplementation in the feed will improve the immunity of the animals, thereby mimicking the immune boosting effect similar that of animal proteins. And also we added nucleotide. Normally, plant-based protein has lack of nucleotide compared to animal proteins. Nucleotide is essential for rapid regeneration of gastrointestinal tract. For example, in the right side, you can see that crypt contains the stem cells, and these stem cells are produced by mitosis and then differentiated and maturated up to the villous tip. When animals are infected with a pathogenic bacteria or there is other viral infections, there will be intestinal damage. Therefore, fillers will be shortened and then crypt will be getting deeper because animals start to produce more cells and increasing mitosis. In that case, they need a nucleotide for rapid generation of enterocytes. When you look at this difference of nucleotide contents between fish meal and soybean meal, especially IMP, inosine monophosphate, alone showed 18 times differences. So we included IMP to the Nextide to compensate those reduction in nucleotides in plant protein source. And we did some in vitro trial with the pig's primary and mouse macrophage cells to test interleukin 10 and interleukin 6. As you can see that effective of single amino acids, methionine and threonine are increasing the anti-inflammatory cytokine production, while tryptophan and arginine decreased pro-inflammatory cytokine production. So, followed by the last slide, we checked the reactive oxygen species ratio and heat shock protein 70 gene expressions with stress situation to the pig's intestinal epithelial cells. As you can see in left side, oxidative stress was significantly reduced in amino acids and nucleotide blend group. And see in right side, heat shock protein 70 gene expression also significantly improved in amino acid and nucleotide blended group. So a proper blend of these amino acids together with the nucleotides will increase the immune defense system in the gastrointestinal tract. So a proper blend of these amino acids together with nucleotide will increase the immune defense system in gastrointestinal tract. So if you look at these concentrations of amino acid contents among our new protein source Nextide and compare fish meal and DPS, which is dried porcine soluble protein. Nextide provide 57% of crude protein and most of them 55 is composed of amino acids. Compared to the major animal proteins, the proportion of amino acids in crude protein is much higher, which means that Nextide contains very less amount of non-protein nitrogen. Lastly, the most important point to use Nextide in the diet is that these amount of functional amino acids are not going to be included as a matrix in the formulation. So these functional amino acids will be added on top of your formulation, thereby providing extra immunity to the animals. I will show you some examples for the functional amino acid trials. A recent study showed that supplementing a functional amino acid blend, including methionine, tryptophan, and threonine, significantly improved growth performance of the pigs after 7 days of salmonella infection, whether they used a low protein diet or high protein diet. For the second study, they found that including functional amino acid blend for the 2 weeks before the actual event of the salmonella inflammation, there was a significant reduction in colonic salmonella proliferation, as you can see in the left side. FAA minus means not including functional amino acid, and FAA plus 0, 1, 2 are including functional amino acid blend on the day of infection or 1 and 2 weeks before infection with the salmonella. As you can see in right side, added functional amino acid 2 weeks before showed significantly improved ADG and FCR, which means we don't know when inflammation challenge happen in the farming situation. So we should supplement Nextide in your diet, which included in this functional amino acid blends before the 2 weeks of the challenge. Then there will be significant protection effects from the pathogenic bacteria. So I'd like to introduce some of in vivo trials by using Nextide and calculate the economic profit. We have much more animal trials data, so if you need more details, then you can contact us. The first animal trial was conducted in TUAF in northern Vietnam. We used 28 days old piglets, and the positive control diet was included 3% of fish meal and 2% of plasma protein, and negative control diet was not included any animal proteins. For the treatment 1 to 4, totally and partially replaced animal protein by 5% of Nextide, as shown in the table. And you can see the feed cost in the right side of the table. Let's see the performance data in the next slide. The gross performance, as you can see, overall ADG, ADFI, FCL were improved trend by replacing animal protein with 5% of Nextide group. Furthermore, treatment 4, which replaced 3% of fish meal and 2% of plasma protein by 5% of Nextide, showed significantly improved even at the lowest feed cost. The gross results suggested that regardless of how much animal protein was replaced, the Nextide 5% treatment group showed relatively improved gross performance. This indicates that when replacing animal protein with Nextide, there was no gross reduction or adverse effect at least. Similar with the gross performance, the best results were observed in the Nextide treatment group for immune indicators. For MDA, malondialdehyde, which is lipid oxidation indicator, interleukin 8 and TNF-alpha, which are inflammation indicator, and immunoglobulin G, which is immunity indicators. Regarding gut health indicators, Nextide 5% treatment group showed a tendency for increasing villus height or significantly improved. The diarrhea rate also showed a tendency to decrease, similarly with villus height when Nextide was supplemented. The second trial was conducted in Gyeongsang National University in Korea. In this trial, we used 24 days old piglets, and for treatments, you can see in the left side. For the gross performance in right side, the Nextide 5% group showed no difference with the positive control, which have 5% of fish meal and 2% of plasma protein for the overall ADG and gain to feed ratio. The meaning of no difference with the positive control is that Nextide 5% group can efficiently replace the whole of animal protein source of the diet. So we can calculate the economic values based on the feed cost with the gross performance. Based on the total cost for a 1 kg of weight gain, Nextide 5% treatment showed 10% reduction of the total cost compared to positive control. And next one, this trial was farm trial. This farm had a commercial diet including 5% of fermented soybean meal, and we replaced it by 5% of Nextide. As a result of this trial, there was about 14% improvement in final body weight with increasing average daily feed intake. Finally, FCL was improved 5.4%. So we calculated economic values. The Nextide 5% treatment increased the feed cost about $2.4 USD per pigs. However, the calculated live weight and live hog price were 6.5 USD, which is much higher than that of increased feed cost. Therefore, the total additional profit from this trial was around 4.0 USD per pig. For the farm with 10,000 pigs, the profit will be increasing approximately 40,000 USD. That's all for my presentation. So I concluded this presentation. Comparative analysis results demonstrated that Nextide contains less anti-nutritional factor and heavy metals, compared with animal protein source. Nextide has probiotic effects with higher thermostability and antibacterial activity. Nextide provides various functions that reducing inflammation, oxidation, and heat stress due to optimal amino acid and IMP composition. As a novel functional protein source, Nextide can safely replace animal protein source via improving feed safety, nutrient digestibility, and immune functions of young and breeding animals. So, thank you for your kind attention. If you have any questions about Nextide, please don't hesitate to contact one of our representatives in your regions. Thank you. Bye bye.